Before I start, I'd like to thank all of the people that contributed to this work. Those in black are the microscopists. Those in blue are the people that make the samples for us, and that can take an awful lot longer than the microscopy, although the analysis tends to take a long time after that. And those in purple are the theorists who are helping us with density functional theory or molecular modelling to be able to understand the phenomena that we're investigating. I've put opportunities for machine learning. This is very much part of the larger plan for the research group. However, it takes time, and it's something that Will is starting as part of his role in my group. So if you get a chance, please do take some time to chat to him or to me, because we'd really like to hear your ideas or have opportunities to collaborate, perhaps. Okay, so I'm a transmission electron microscopist. This is a scanning transmission electron microscope. Hopefully most people in the audience are vaguely familiar with a scanning TEM. Fantastic. So this is the one in Manchester. And I just wanted to highlight that most of the images I'm going to be showing you are these high-angle annular dark field images. So they're really powerful because particularly when you're looking at atomically thin samples, you can interpret them very directly because increase in intensity in this image is a sign of a higher atomic number, roughly z squared or z to the 1.7. So, for example, in this tiny little nanosheet of molybdenum-doped tungsten oxide, we can see which are the tungsten atoms and which are the molybdenum atoms just from the intensity of those atomic positions. I also wanted to highlight on this slide that one of the great things about doing scanning transmission electron microscopy is that for every pixel you can also collect other information. So you can also collect the electron energy loss spectrum, which can tell you about the chemical states in the material, and you can also collect the energy dispersive X-ray spectroscopy data, which is telling you about the elements that are present within that sampled region. Okay, so in this talk, I wanted first of all give you an exemplar of some work that we're doing. I'm from Manchester. Quite a lot of what you're seeing is related to two-dimensional materials in some way or other. It's not actually in the job description, but it is still a big part of the work that goes on in Manchester. And I have about half my group focused in the National Graphene Centre, National Graphene Institute, and on the other side of the road, we have the Electron Microscopy Centre. So it's very much a collaboration between those two centres. So, as I say, we'll start with an example of some work we haven't done in liquids, what we would like to study this particular reaction in liquids. We'll then move on to how we can do TEM imaging in liquids. You'll all be aware that generally transmission electron microscopy is done in an ultra-high vacuum. And then I'm going to show you how we can use 2D heterostructure liquid cells, that's liquid cells built from graphene, in order to be able to interrogate some of these chemical reactions that we're interested in. Okay, so to start with, I don't know if many people are aware about this class of materials which are called clays or micas. They're aluminosilicate structures, so they have these layers that you can see up here. So these are tetrahedra and octahedra of oxygen, aluminium, and silicon. And they create these long layers, and sticking them together are these exchangeable cations. Now, these materials are ubiquitous. They are all over the place. You'll find them in filtration membranes in fuel cells. They are very important for understanding radioactive waste. I first came across clays, particularly illite, working with a collaborator who wanted to understand what happens when there is, like the Fukushima natural Fukushima radiation hazard, and there are radioactive ions that leak into the soil. You need to understand whether those radioactive ions are going to get into the water and how far they're going to penetrate through the soil. That means you need to understand how an ion interacts with soil, and most of the soil is made of clays and micas. So we need to understand how the ions that are present in a solution are going to interact with a clay or a mica. We'd also done some collaboration with a colleague of mine, Marcelo Hidalgo Ladade, and he had found that when you make clays and micas, not bulk powders, but down to a single atomic layer thick, they behave very differently. So the goal of this project was to understand what happens when you put a clay or a mica, which is atomically thin, into a solution containing a different type of cation. What happens? Okay, so this is an image, it's a cross-sectional imaging. I'm sorry about the projector, everything has gone really red. It's not as red on my screen, and there seems to be nothing we can do about it. So you're just going to have to look at images that are a little bit pink saturated, and I'm going to apologize for that. So what we can see here is the cross-sectional image. This is our vermiculite crystal. Here we have two layers of vermiculite, and sandwiched between these two layers of aluminosilicate is this layer of cations. Now, Marcelo wanted to look at proton substitution. However, electron microscopy isn't very good at looking at very light ions, and so we said, well, could we do cesium? And he said, yeah, you can do cesium. This also exchanges, and in fact, cesium is a very important ion for radioactive contamination. So in this image, you can see the cross-sectional view of the aluminosilicate layers stuck together with this layer of cations. Here we have three layers, so you have two cation layers. You can also look at these samples in plan view. So now we're looking down onto the planes of these ions, and we can see the hexagonal arrangement of the substituted cations stuck in the middle of that sandwich. If we have two layers and we stick them on top of each other, these are slightly offset, and so we end up with these linear features. Okay, so we understand that we can see the ions when they're in the aluminosilicate layers. We want to understand what happens when you have an atomically thin crystal. So to do that, we had to take over 60 samples looking at different thicknesses of the aluminosilicate, so different thicknesses of the material, different solution chemistries, different times in the solution, to be able to try and understand the dynamics of a liquid phase reaction. And we're now going to interrogate these. We're roughly going to estimate how much of these bright cesium ions are present within these images, understand how far they've penetrated, so we measure the penetration distance along each layer, we measure that as a mean, and then we calculate how far the ions can penetrate into an average layer depending on the thickness of the material. And after several years of work, what you end up with is this rather small graph here, which is telling you that there are 5 orders of magnitude difference in the speed with which ions can penetrate through this aluminosilicate space if your material is only 2 atomically layers thick, as opposed to if it's much thicker or indeed bulk. And for the purposes of this behaviour, bulk is now just 10 atomic layers, so 10 layers of aluminosilicate. Okay. So we were very pleased with this. We found that it also works not just in a TEM, it also works when you make a material which is made from few layer flakes. You put it into the membrane, and you put the membrane into the solution, and you get the ion exchange at orders of magnitude in difference in speed. So we submitted this, we were very pleased, and the reviewers said, okay, but why? So we said, well, good question. So we did some atomic force microscopy in liquids, and this is multi-modal, you see. So by looking at the clay on a surface in liquid and dry, we can measure the difference in that height. And what that's telling us is how much of the water can get into that interlayer space and expand it. And this is quite a challenging experiment. You have to make sure that your layer is really firmly stuck to the surface so that none of that water is going underneath that layer and lifting it up. But we've done that because you can see here that when you only have one aluminosilicate layer, one crystalline layer, then it doesn't change in height at all when you put it into the liquid. However, when you have 2 layers, that space between the 2 aluminosilicate, the interlayer space, increases the height of the sample by 5 nanometers. That is just enormous. But it is still a perfect crystal. When we have 3 layers, so 2 interlayer spaces, now the increase in height is only 3 nanometers, and that's spread over 2 interlayer spaces. So there's a massive difference in the ability of that crystal to swell the interlayer space when you have a very small number of layers. And that's due to the potentials interactions, which was modelling done by Francois Peter's group in Antwerp. So you can see this swelling is very heavily dependent on the number of layers, and that effectively means that this 5 nanometer space, and this is going to become important later, this 5 nanometer space, the ions behave as if they were in free water. So when you have a space of 5 nanometers, the ions that are diffusing into that space have the same speed as they would diffusing through free water. You can also now look at these materials and understand the surface ions, because we saw this swelling of the interlayer space, but when you make a material just a few layers thick, then most of it isn't interlayer anymore. Most of the positions that the ions can exchange onto is actually the upper and lower surface, and this is a real challenge for electron microscopy. Yeah, go on.
Was that swelling normalized per layer?
Yes.
Okay, that makes sense.
So here it's normalized per layer, this data here. Swelling per interlayer. Brilliant. Okay, so now we want to look at the surfaces. Now, this is really challenging microscopy. Clays and micas are very beam sensitive. They typically would only survive a few images at atomic resolution. When you make them a single layer thick and you put them into a microscope, you have to keep them wet, because the ions on the surfaces are hydrated ions. So that is a challenge. You can do it, as it turns out, by putting them in and being really fast, or you can put them in and put graphene on top, which I'll tell you more about later. So this is imaging of surface ions on a single monolayer. Now, no interlayer space, so all the ions you can see are on either the top or bottom surface. Interestingly, what we found is that either they're all on the bottom or all on the top surfaces in general. We don't understand that, if anyone's got any ideas of why it is, but the exchange on the surfaces tends to completely fill one surface and then completely fill the other. No preference for the top or bottom that we could see within this data. So this is a cross-sectional image through a 3-layer thick sample. Cesium is exchanged on the bottom and we have potassium exchanged on the top. You can see they have slightly different heights relative to the underlying clay or mica, and we can also see that they take up a slightly different position on the surface. So they aren't directly substituting the cesium for the position of the potassium. The cesium is going into a different site. And that is because cesium is known to have a more hydrated, a more solvated structure than the potassium. And that means that that solvation shell allows the ion to have a different preferred lattice location. And these are images that are being used to investigate this. Potassium is much lighter. It's much more like the aluminosilicate lattice. So we need to use a different imaging mode. We're using the annular bright field imaging mode, which is more like a traditional bright field TEM image. We can't see the position of the potassium in the annular dark field image because it just gets lost in the background of that aluminium and silicon. Okay. So this is a really challenging problem. So we have done this for one image. It took the researcher several months. There is huge potential to be able to study these types of noisy images and to be able to correlate the ADF and the annular bright field image, because we need to understand where these cesium ions are with respect to the underlying lattice. And we can only see the underlying lattice in these images for the annular bright field, and we can't see the cesium then with anything like so much clarity. In fact, it's even more complicated than that. The best focus that you get for the annular bright field image isn't the same as the best focus for the annular dark field image. And so we have to take two pairs of images, one where the annular dark field is in focus and one where the annular bright field is in focus. And there will be an average. The atomic positions that we find for those two images broadly appear to be the same, but there's big error bars in our understanding of that behavior. And again, if anyone would like to help us to interpret those types of data, that would be fantastic. So those were snapshots taken with laborious TEM information, and we still haven't really been able to probe the motion, the behavior of how those ions are penetrating. We can get you the mean diffusivity speeds, but we can't get whether the effect of stacking faults is increasing the rate of ion diffusion. Is the effect of different surfaces changing the rates of ion diffusion? We'd like to be able to look in the TEM and see that type of behavior, but that requires doing that chemical process inside the TEM. Okay, so we're just waking up now. So this is examples of some pictures of horses that were painted back in 1790, 1870. The importance of the dates is that they are before photography. And before photography, people took or painted pictures of horses which were a little bit like Superman, so flying with their front hooves and back hooves both off the floor at the same time. Now, with the present era of photography, you can take slow motion images and you can see that the horse never does a Superman impression while it is galloping, however fast it is going. Please, nobody ask me questions about horses. I have no understanding of horses. So the take-home message from this is that in order to be able to understand the detailed mechanisms of a process, it really helps to be able to image the different stages and image the same thing at different stages, which is what's special in the TEM. As I said, we're not looking at horses, and for the purpose of this talk, we're interested in processes that happen in liquids, and that's a huge range of processes. So that could be chemistry, the ion exchange I've already talked about. We're also interested in investigating electrochemical reactions, wet synthesis of nanostructures, which I'm going to talk about a bit at the end of the talk. Lots of things in physics, and a lot of our colleagues in the National Graphene Institute are focused on understanding the fundamental of fluid flow through confined spaces, which again I'll come on to later. Again, in materials and biology, lots of things happen in liquid. Very little interesting stuff happens in perfect vacuum, with the exception perhaps of quantum computers, so sorry. Fantastic. Okay, so you can imagine we've moved on from horses to fish. Now the experiment that you want to do is represented by this fish. And we want to put it in the TEM and study that fish being fed or interacting with another fish. So if we put it into the vacuum of the TEM, in the best case scenario, you have the goldfish, which is put on a plate, and in the worst case scenario, it's completely dried out and really bears very little resemblance to that solution process. And this has been shown quite widely in gas cell microscopy. So it's easier to get a gas into the electron microscope than it is to get a liquid, and I'll be happy to talk to you about that afterwards if you're interested. But it had never been shown really for liquid that there is a difference in the behaviour or the atomic arrangements for a material when it is in a liquid as opposed to when it's in a vacuum. We can also use cryoelectron microscopy, and sorry about the cryoelectron microscopy is a fantastic technique. Nobel Prizes, science and nature papers coming out all the time. It's incredibly powerful. But hopefully cryoelectron microscopists would agree that it is not the same as looking at your liquid phase process happening in real time. So what we want to be able to do is create a tiny Petri dish that we can put into the electron microscope, and we can do that with conventional holder systems, which tend to be based on silicon nitride, but they compromise the spatial resolution and the elemental analysis capabilities, as I'll mention in a minute. And so our approach is to make this tiny nano aquarium inside the microscope and to use graphene to be able to do that, or other two-dimensional materials. Okay, so I mentioned that there's a variety of commercial systems, and we're really fortunate to have a full range of the protochips, liquid cells in the electron microscope that we have in Manchester. So the holder that goes into the side of the microscope looks something like this. There's a little pumping station that goes on the side. If you take a cross-section through the end of that TEM holder, you'll see this narrow channel of liquid. Actually, most of the liquid goes all the way around the outside of that region you're able to image with the electron beam. And this separation of the amount of liquid that you get between those two windows is in the best case 100 nanometers, and in most cases a micrometer. That's a problem because it seriously degrades the resolution that you're able to achieve. If you're doing TEM, the first thing a microscopist might ask you is your sample less than 10 nanometers thick? And if you say no, it's less than 100 nanometers thick, please. And if you start with a liquid layer that's over hundreds of nanometers thick, your ability to do atomic resolution or high resolution analysis is vastly decreased. And as I say, just the geometry of that liquid cell holding the liquid together inside the nano aquarium often reduces the amount of information you can get using EDX spectroscopy, because it blocks a lot of the X-rays, and it also degrades the signal you get with electron energy loss spectroscopy, because all the electrons have mostly interacted with is the water. Okay, so these are some, again, not my work, this is some pioneering work which has come through, but I'm just using it to illustrate the sort of the conventional images that you might expect to see with a standard commercial liquid cell. So really powerful. They allow you to look at nanoassembly processes, they allow you to look at electron beam deposition processes, but they tend to be at a resolution of around the 5 to 10 nanometers, which is what's shown here. And getting atomic resolution is really, really challenging. Okay, so back in 2010, the Lawrence Blerke group, managed to demonstrate that you could take two sheets of graphene and just bring them together in the presence of liquid, and you could trap this tiny volume of the solution that you were interested in, and you put that in the microscope, and that was a tiny graphene liquid cell. This generated huge excitement in the field at the time. I was just starting in Manchester. Everyone said, we must have graphene liquid cells. They're fantastic. And they have achieved some really powerful results. However, having tried to work with these type of graphene liquid cells, I don't know if anyone has tried to carry soup with cling film. No? There's a reason why people do not try and take soup around. You don't take your soup to the lab with cling film, do you? Because it leaks out of the creases. And that's effectively what you're trying to do when you have a graphene liquid cell made from two sheets of graphene. You have incredibly flexible, thin membranes, and you have a solution that you're trying to trap between them. And so most of the pockets are too big, most of them leak, and finding a liquid layer that is the right thickness is a bit of an art. So although the sample prep is in theory quite straightforward, in practice there's only been around 20 different papers that have come out almost entirely from this research group using graphene liquid cells to do reactions, and all of those reactions have been beam-induced processes, or looking at the native solution, the properties of the native solution. So we wondered, could we improve on this process? And as I said, I work in the National Graphene Institute, and a lot of my colleagues asked me to spend a lot of time looking at heterostructures. Now, 2D heterostructures, in case you're not aware, is when you take the zoo of two-dimensional materials that has been discovered over the last 2 decades, and you combine them to produce a new electronic structure. So you might decide that you want to combine a conductor, graphene, and then an insulator, boron nitride, and then a semiconductor, monolayer molybdenum disulfide, and then another insulator, and then another conductor, and that creates a light-emitting diode or a transistor. For the purposes of this talk, I'm also going to mention some of the work where you don't have to combine in this heterostructure stack complete layers. If you pattern gaps into your two-dimensional material, then you create regions that you can fill with a liquid or with a gas. And that nano-channel work is a big area in Manchester. And so that's the nano-channel that you can see in the middle there. Okay, so we have, as I said, done lots of work in the transmission electron microscope using this encapsulation of our two-dimensional materials that are unstable in air. So this is chromium tribromide, which is not stable in air, and this one is tantalum disulfide, which actually, when you encapsulate inside your device, improves over time, as opposed to most of them that just fall apart. I'd be happy to talk to you about this. This one particularly, we did some EELS analysis on the degradation structure and found that the oxygen, which is part of the trapped species that is within the layer and is able to diffuse inside, exists in both a crystalline form and an amorphous form using principal component analysis. And so the fact that you have these two forms tells you that there's not just rubbish trapped between the two layers that are encapsulating your two-dimensional material, that oxygen is actually being incorporated into the lattice. Right, so we have this platform. As I mentioned, Andre Geim and Radha Boyer's groups in Manchester are also really interested in using this platform to be able to look at the process of flow for liquids and gases. That's really important in a huge range of processes, not just in batteries and fuel cells, so material science type applications, separation membranes. Again, Rahul Nair does a lot of work on desalination, looking at the diffusion of salts through these very small gaps between two-dimensional materials and nanochannels. Now, prior to the work starting in Manchester, a lot of this had been investigated using conventional electron beam lithography approaches on silicon and silicon nitride. The problem with that is if you're looking at the effect of nanoscale confinement on the flow of a liquid or gas, and your material has a surface roughness of 1 to 10 nanometers, then your ability to probe what is happening when you have a gap of 1 to 10 nanometers is very low, because you're dominated by surface roughness effects. Now, as you'll know, two-dimensional materials have the advantage that they are atomically flat, and so you have a very well understood interaction with the surface. So this is the type of device that we have been looking at for around about a decade now, where we were looking at flowing the liquid through this nanochannel region. There's top and bottom graphene layers, and what the the collaborators were interested in was trying to characterize these flow channels. If we zoom in the TEM, you can see this channel really is empty. Once it's in the TEM, it's empty. And this is the spacer layer with the top and bottom graphite layers. When you look at the flow of the liquid out through these channels, and you plot that as a function of the channel height, so that's n number of layers, which means that this is 1 nanometer height here, you can see that there is a vastly increased flow rate when you have a height of just 1 nanometer, and then again it starts to go up to the bulk as you start getting it out to 5 and then 10 nanometers. And you can understand this based on an increase in the order of the water in between the two graphene sheets, which means that you get much faster flow. So effectively you have a higher pressure, which gives you this much faster flow through these channels. Now the important take-home message here is that you do get significant fluid flow through nano channels that are made from two-dimensional materials. Should mention more recently, we've been working with them to characterize their nanochannels for gas flow through the materials. And here, it seems to me to be very surprising, but is clearly demonstrated that surface roughness, even molybdenum disulfide, because it has sulfur atoms that are sticking up, if you're a gas molecule, that is rough, as opposed to graphene, where all the carbon atoms are in a nice plain layer. And so you get a different behavior when you have sticking up surface of sulfur atoms. You get this a diffuse type of scattering, so a more conventional scattering process, as opposed to when you have graphene, when you have this specular flow, so you effectively have like a billiard board bouncing off between the layers. You get very fast flow through graphene and hexagonal boron nitride. Anyway, sorry. So, bringing back on topic, we're looking for liquid in the transmission electron microscope. So we're going to take one of our two-dimensional crystals, we're going to use electron beam lithography to pattern a well that is the right size to take the right amount of liquid that we want to be able to use to do our experiment. So now you're taking your soup home in a Tupperware. I don't know what Tupperware is in American. Do you have Tupperwares in America? Oh, great. Okay, so you've got your Tupperware, and indeed, when you have a Tupperware, you can see the position of the individual atoms. This is a platinum salt within the graphene liquid cell. You can see that this is the size of the cell, but it's lithography. You can choose whatever size this cell is. The filling is better when you have smaller cells. If you start to make your cells tens of microns in size, you end up with them filled around the edges and the graphene sheets stick together in the middle. Okay, so we did some imaging on these, but what you'll notice is this isn't atomic resolution imaging dynamics. And that's because if you image these cells at atomic resolution, you're looking at one of the graphene windows, and even at 80 keV, you will create damage to that window. And when you have damage to an atomically thin window, your liquid escapes. So we had to come up with an alternative to be able to look at the atomic motion within these graphene liquid cells. Just as an aside, you can do the elemental mapping. Elemental mapping we expected to be as good as when you didn't have any graphene, because you've got effectively nothing to block the X-rays. However, it turns out it's better than when you have the same sample imaged in vacuum. And we put that down to the fact that the solvation shell around our nanomaterial is actually helping to stabilize it. So you get less electron beam damage, you get the heat that's being generated by the electron beam is more easily dissipated, and so you get less damage. You can use higher electron flux in order to be able to image nanoparticles and take elemental maps in liquid. That's going to become important later on. Okay, so in summary, we have our conventional silicon flow systems, which do create some really useful stuff. We have the more traditional graphene, and then we have our lithographically patterned graphene cells. And you don't just have to put one layer. And in fact, we had a paper with Andre Geim's group a few years ago, where they got an efficiency of a light-emitting diode that was not sufficient to get it accepted into Nature Nano. And so they said, okay, Fred, go away and make 4 of them and stick them on top of each other, and we'll measure the efficiency of that light-emitting diode. And so we can go for the same approach here, is that we have one graphene liquid cell before, and now we're going to just put another one on top. We're using this stacking approach. All of this is done in using single crystals, which are exfoliated, mechanically exfoliated. That's using Scotch tape and pencils. You peel them layer by layer. You use a micro manipulator and you stick them back down together. Only in our case, we stick them back down in the presence of a liquid. So if we stick back together 2 pairs of liquid cells, you end up with this structure that you can see here. And why is this interesting? It's interesting because you can now use the electron beam to drill a hole in that separation membrane, and you can make the 2 liquids mix under the electron beam. And that was not possible, or is not possible, with any conventional liquid cell technology. You can have 2 flows, and you can flow in different liquids, but they will mix in some random position, most likely miles away from the region that you're able to image. Miles in a microscopy sense. Okay, so this is the broad geometry. The liquid cells that we have don't just have one nano aquarium. We're doing electron beam lithography, so we would typically pattern 100 by 100 individual wells within these crystals. So we can repeat the experiments at different electron beam dose rates, and we're working on opportunities for being able to repeat those experiments with different conditions. Okay, so we drill a hole in our separation membrane. In this case, it is molybdenum disulfide. We can then create a crack. Again, apologies for the quality of this image. There is a crack that is going through that separation membrane, and that cracking means that you get widespread mixing of the 2 liquids. That's the crack at the end. The total thickness of these graphene liquid cells is 70 nanometers. So typically 35 nanometers on the top, 35 nanometers on the bottom. So you're still in the regime where we can get nice atomic resolution imaging processes. To demonstrate this technique, we chose to look at calcium carbonate precipitation. So calcium carbonate precipitation is really important. It plays an important role in corals and the growth of marine organisms, and understanding how marine organisms are going to change their growth rates as a function of different pHs is really important when we think about global warming, which is changing the pHs of the oceans. There's a wide range of different issues that can change how calcium carbonate precipitates, but it's a very complicated and very poorly understood process. So we decided we'd have a go. It's a relatively simple mixing reaction. We can mix together calcium carbonate with sodium hydroxide with calcium carbonate. So we're going to get here, what's that? Yeah, so we've got, calcium chloride with sodium carbonate, and we end up with calcium carbonate. Okay, great. So that's the hole that we've drilled in the middle. I'm going to make that video replay. Oops. Okay, so if you keep your eye on this, you'll see it's looping. But the first thing that we saw when we looked after we drilled this hole were these big bright globules. Okay, and that isn't what you'd normally expect with a precipitation reaction. Precipitation reaction, you expect to form little clusters and for clusters to grow. And that is the conventional precipitation route. But what we see was these dense liquid globules, and we didn't understand at all what was going on. So we got in contact with Denis Gabauer, who's been studying this for several decades, and he was very excited and said, this is a verification of the liquid-liquid phase separation theory. So you have these dense liquid globules which are able to form the first stage of a binodal decomposition, and then the denser calcium-rich liquid regions will stick to the window, and then it's them that start forming these disordered clusters, and then the disordered clusters gradually densify and form the crystals. Okay. Not going to go into this, except that all of that mapping of crystals, which is done here by doing a size analysis, this is all manual. I'm sure it could be done much better. But looking at the manual size analysis actually misses the key parts of this reaction, which is that denser liquid phase. And because the electron beam is dehydrating the system, changing the system in many ways, in order to form what you would recognize as being a calcium carbonate crystal, these facets tell you it's a nice single crystal. In order to form that, you have to turn the electron beam off and do the reaction with the electron beam off. So you break the membrane or pierce the membrane, allow the liquids to mix, and now you do the snapshot approach. So you look at it after 5 seconds, after 10 seconds, after 15 seconds. If you just keep looking at it, you never go beyond these nano-sized clusters, which we assume is due to dehydration. We would love to be able to get to being able to use lower electron doses and interpret the images in a similar way. Again, opportunities there for machine learning. Okay, how am I doing for time?
15 minutes.
Oh, super. Okay, so The same experiment means that we have a suspended crystal in between two layers of liquid. And remember I said before, if you do atomic resolution imaging on a surface, you create point defects, you create vacancies which allow the liquid to escape. However, if you have another liquid pocket on the other side, what happens is that the liquid just mixes around and you don't end up losing the solution chemistry of your system. And so that's what's demonstrated here. This is the atomic resolution imaging done on the top graphene layer, the middle molybdenum disulfide layer, and the bottom graphene layer. And you can see the lattices and the atoms if it wasn't quite so pink. Sorry. Okay, so these are the images that you get when you are observing the middle separation membrane. That's the molybdenum disulfide layer, and you can see the salt crystals have precipitated out these individual platinum atoms which are stuck to that molybdenum disulfide surface. Now this is the same behavior observed in a vacuum. You can recover this same behavior either by leaving the electron beam on one of the graphene windows and allowing all the liquid to escape, and you end up recovering this behavior. Or you can do the experiment just on a single layer of molybdenum disulfide, drop some platinum salt solution on the surface and then put it into the microscope. And when it dries up, you recover this same behavior. Okay, so you can see that although the signal to noise ratio that we're getting here is not as good as we get when we have a vacuum imaging, we're now able to take hundreds of images of the solution, and we have tracked the behavior for those hundreds of images, and when we compare the behavior of the atoms in the atoms in the first 10 and in the last 10, we don't see any differences in their behavior. Okay. So we want to be able to now dig in and get information on the location of the individual atoms with respect to the underlying lattice. And this is the only technique that can do this. Liquid AFM can get information on individual surface atoms surrounded by liquid, but it can't tell you very much about the dynamics of that behavior, and it can't tell you anything about the underlying lattice that the atom is sitting, so that you can get the preferred lattice sites. So here we can do an analysis to look at the images and understand by a pattern matching process, understand where the platinum atoms prefer to sit. So you can see that you get platinum atoms sitting over a molybdenum site in the molybdenum disulfide, over a sulfur site in the molybdenum disulfide, and over that whole site in the molybdenum disulfide. And rather heroically, Nick has used conventional pattern matching approaches to analyze many data sets. So we have 40 data sets, they all have hundreds of images, and this is incredibly time consuming to analyze this type of data. But with 90,000 atomic measurements, we can now compare the preferred positions for platinum atoms on molybdenum disulfide in an aqueous salt solution as opposed to in the vacuum environment. And this type of behavior had been observed before because these are hydrogen evolution reaction catalysts. They are, have been widely studied by vacuum phase TEM, and the understanding was that all of the platinum sits above the sulfur sites. However, when you study them in the liquid solution, which is much more like the environment that they'd actually be doing the catalysis, we find that the platinum atoms are almost equally likely to be sitting above a molybdenum site as they are above a sulfur site. And that's because in the vacuum phase TEM, the behavior of the platinum is overwhelmingly dominated by sulfur lattice vacancies, and they're generated in the very first image that you do in the electron microscope, and then the platinum tends to stick on those sulfur vacancies and move with them or hop between them. And if you look at this behavior, it very closely matches what you'd expect by density functional theory. Okay, so you can then not just look at the preferred atomic lattice sites, you can also look at the dynamic motion of the platinum. I'm not playing. Yeah, so you can pull out these pretty models just to illustrate what's going on. So these red atoms are the platinum lattice. The blue lines are where they are moving over the surface, and then underneath you've got the purple and yellow, which is the underlying molybdenum disulfide atoms. For this analysis, we've neglected anything where the platinums start to interact with each other or where they start to get to the edge and there's some defective regions. We're really only looking at the behaviour of the platinum atoms on a pristine surface. Okay, and we have tried to look at the effects of different electron doses, because clearly the electron beam is having an effect on this system. It's a little bit like trying to understand how somebody would swim by asking them to go in a boiling paddling pool and throwing things at them, and then trying to understand how somebody learns to swim. And so we really would like to have a less interaction of the electron beam with the system. With other experiments, we've had a lot of success by looking at different electron doses and being able to extrapolate back to what would happen if there was no electron beam. Or getting to a point where your dose is sufficiently low that the electron beam processes have time to relax back in between each image, so you don't see any change in behavior cumulative over time. Now, in these systems, we do see damage to the samples, and we are limited in how low we can go in the electron flux. And that's because we still want to be able to get the positions of those platinum atoms, and we want to be able to see the location of the underlying molybdenum disulfide atom. So we've got about an order of magnitude difference in the dose that we can use, but we'd really like to be able to go much further down in terms of electron dose. If we look at the diffusion behaviour, we can see there are really big differences between the wet and dry processes. It's much slower in the dry condition, as you would expect, and the electron dose has a smaller effect than the wet versus the dry. Interestingly, if you reduce the electron beam dose, the atoms move more quickly. That means that we aren't looking at an electron beam driven motion of the platinum atoms. They are moving more quickly when we put less electrons into the systems, and therefore we propose that the electron beam is actually slowing down the systems by creating ionic species that interact with those atoms moving around on the surface. Okay. So what I've talked to you about so far is all two-dimensional, and you're going to hear some lovely work about three-dimensional microscopy later on today. I'm just going to give you a little brief introduction to some of the work that we've been doing in that space. Many of you may be aware of the use of cryoelectron microscopy to create three-dimensional reconstructions of proteins, and this is the single particle reconstruction approach. And the single particle reconstruction approach is extremely badly named in my opinion, because it is anything but a single particle that you are studying. What you are typically studying is millions of individual proteins or objects, whatever it is that you want to study, and you are assuming they are all the same. Okay, and that is, even in biology, that's not a very good assumption in lots of cases, and there's some very clever work to segment and work out which of the different classes within the different protein structures to understand different conformations. In physical sciences, single particle reconstruction isn't very widely applied. And I think, in fact, one of the few approaches where it has been really successfully done is when, by the the Berkeley group, who have looked at these graphene liquid cells again. And in the graphene liquid cells, they've had the same platinum particles that you saw back from the 2010. But now they have done some very clever analysis of those platinum atoms and use the fact that because they're in the liquid, the platinum can rotate, it can roll. And you therefore have a lot of different measurements of that platinum atom at atomic resolution for different orientations. And you can then process all those different orientations to create a tomographic reconstruction. You have to make some assumptions that nanoparticle isn't changing in the solution, but still, this is the paper that they published back in 2020, showing reconstructions for a few platinum nanoparticles. Okay, so what we first came to trying to do single particle reconstruction when we wanted to start including elemental analysis in our conventional tomography. And you're going to hear later, but conventional tomography, you take your sample and you don't let the liquid rotate it, you manually use the holder in the TEM, you tilt it at different angles. This is in some ways easier because you've got a better understanding of what your tilt angle difference might be from one image to the next. If you've got a particle randomly rotating in the liquid, it could randomly rotate in all different directions. It certainly doesn't choose to go the same way each time. However, that still requires a relatively high electron dose, and that is also true in a lot of tomography. We are limited, even for imaging, this is an image of lots of different nanoparticles. You can take one of these nanoparticles, you can rotate it many different angles, and you can end up with a three-dimensional reconstruction. I'll show you that in a minute. However, if you want to take an elemental map, elemental mapping takes orders of magnitude more electrons to create enough X-rays to create an elemental map. So that means we need now to go to a particle that is more robust, or if we want to still be able to look at the same interesting catalytically active nanoparticles, we need to find a different way to perform a three-dimensional reconstruction, including elemental information. So our approach to that is this single particle reconstruction. Single particle reconstruction, in this case, is Yiqi Wang's PhD. So he took 1,000 nanoparticles from images like these. He segmented them. He measured the elemental composition, that you could get the mean elemental composition by looking at the EDX signal for that nanoparticle within that image. He then found that varied between 40% and 70% platinum. These are all, as I said, platinum nickel nanoparticles. He then looked at the mean size measured in projection and found that was approximately 20 nanometers. As I said, single particle reconstruction requires that your particles are all the same. And so when he tried to reconstruct this data using all 1,000 nanoparticles, it didn't work. And so you had to down select through the particles, and his approach was to say, okay, well, I'll just take the nanoparticles that have a composition of roughly 60%, and they have a size which is a little bit tighter. We'll throw away the outliers in that case as well. And when you do that, you find that you can use the information in the simultaneously obtained images, but take the chemical information that was acquired at the same time. This chemical information hasn't got enough detail in it to tell you the orientation of the individual particles. But because they're simultaneously acquired, you can just take the registration information from the high-angle angular dark field, use the elemental maps, and combine them to get an average elemental map for that nanoparticle section. And we have confidence in this result because if we, instead of taking the 55 to 65% nanoparticles, we take the 45 to 55% nanoparticles, we get a different reconstruction, but it has the same broad chemical segregation that I'm going to show you in a minute. So we are confident that this is a representation of the full nanoparticle population, even though we've had to only choose a subset of the nanoparticles to perform the reconstruction successfully. So this is just some details of how we are then trying to understand whether the orientations that we've got are sufficient to cover the orientation space. And the reconstruction that we obtain is shown here. We aren't really at atomic resolution. What we have done here is create an atomic model because we know the size of the lattice. We know the morphology of the particle with a resolution of about a nanometer. And then we filled that with platinum and nickel based on the voxel concentration that we know for that nanometer by nanometer square region. In theory, you'd be able to do this type of analysis at higher spatial resolution, but you'd need more electron dose, so you need more stable particles, or indeed you just need to measure more particles. Okay, this is quite a complicated elemental distribution. I don't need to go into the details of that for this audience, just to say that we've been working with the chemists more recently to understand how this can change the catalytic performance and how you can form these nanoparticles, which is a liquid phase reaction going from an original rhombic dodecahedral particle to this very faceted concave surface shape. And that is caused by an oleamine solution which etches out of the nickel, and it gives you a doubling of the specific activity for these types of nanoparticles, and they're also really stable. So this is a really exciting way of synthesizing robust platinum nickel nanoparticles, again, for the hydrogen evolution reaction. Okay, I think in the interest of time, I'm not going to go through all the details of this last. You want 2 minutes, all right? Okay, 2 minutes. We'll go through this very quickly. That was single particle reconstruction. There's a different approach to be able to get three-dimensional information, and I think you've probably already heard from Angus about a variety of different indirect methods for obtaining three-dimensional information. So I thought it might be interesting for this audience just to see a slightly different indirect reconstruction approach. Now, Again, I'm not sure how familiar people are with convergent beam electron diffraction, but essentially what you're looking at here is a convergent beam electron diffraction pattern for graphene. So it's hexagonal, it reflects the hexagonal graphene lattice. If you have in your material a layer of graphene and then a layer of either another graphene or a hexagonal boron nitride, you end up with two sets of hexagonal spots overlapping with each other. Okay, now this is a phase contrast and interference technique. And so you're going to get interference at the overlap positions between these two types of disks. Okay, and that is going to be varied depending on whether you have a small twist orientation between your hexagonal boron nitride and your graphene, or you have the same, perfect alignment, in which case you just get slight differences in position because the hexagonal boron nitride has a slightly larger lattice constant than the graphene. So you're going to get interference between these. And indeed, you get beautiful patterns in your convergent beam electron diffraction patterns which show you the interference fringes. So when your material is the perfect orientation, so no twist between the layers, then what you have is one lattice that's slightly larger than the other. And so your interference lines are going to be in the outer direction away from the zero order spot, as opposed to when you've got a slight twist where you have both an overlap which is slightly larger and an overlap of twists, so you end up with slightly different orientations. So this is the experimental data, and this is the beautiful simulation work that Tatiana does, and this is the paper that we published together, and this is the review article on that approach, going into lots of detail into the theory of that reconstruction. This is the last slide. So you can't just understand the stacking and the twist angle with convergent beam electron diffraction. It's very sensitive to the separation of those two layers and the precise information within each of those layer structures. And so you can get three-dimensional information on rippling and on objects that happen to be sat on top of the graphene. And again, this is showing you simulations and theory for how these convergent beam electron diffraction patterns are able to pull up atomic scale deformation of those layers, and indeed will be able to tell you about information that is in liquid that is trapped between those two layers. Why do we want to do this rather than tomography? Well, convergent beam electron diffraction inherently requires far fewer electrons to get an interference pattern than it does to obtain the direct image. Okay, so in conclusion, I hope I've shown you that 2D heterostructure liquid cells are an exciting playground to be able to maybe do some experiments that we didn't think that we could previously do within the electron microscope. I hope I've shown you that there's an awful lot of things that we would like to be able to do with the images that we obtain that we're still not able to do using conventional techniques. I'd like to thank all of my research group and collaborators for the efforts putting this together, for the funders, and of course you for your attention. Thank you very much indeed.